Good morning, ladies and gentlemen. My name is Andreas Zumbeuhl, and you probably see that is not the name that is in the program. Unfortunately, 2 out of 3 speakers were sick or didn't get a visa, so we had to do something with the session. The middle talk is the same, but the beginning and the end will be slightly different. So, I step in as the first speaker and we'll talk about mechano-responsive drug delivery. And here we start with a continuation of the first session this morning. In the big hall, there was cardiovascular disease. And it's a disease we all know. We all know people that died of a heart attack. And maybe you asked yourself a couple of times already, do you have a prevalence for a heart attack? Will I not finish this talk and fall down dead? It's a very, very strange disease because within seconds you're gone, and it's very hard for the relatives. It's a very prevalent disease. It's the most prevalent cause of death in the world. And for Switzerland, it's quite surprising. In the past 35 years, the data haven't changed. We have less death, but still 50% of all people die of myocardial infarction. Now, from the time where the patient has a myocardial infarction, it takes about 1 to 2 hours until he or she is in the high technology environment of a hospital and can be treated. And there's a real gap within these 1 or 2 hours, and we'd like to bridge that gap. Now, myocardial infarction is obviously a disease of the blood vessels, and it's very much a physics disease. If you look now at the stenosed artery, what you can see is we change the physics of the blood hemodynamics. You have to squeeze a lot of blood through a narrow artery, and there's a chance here. Obviously, when you have a lesion here, the artery is blocked, and then the rest of the heart is not perfused with oxygen anymore, and this causes necrosis. And the necrosed tissue will never grow back. Now, our idea is to really use the changes in shear stress in the stenosed artery to release a drug at the site where we need it during these 1 or 2 hours that it takes to take the patient to the hospital. Now, physics is on our side. A typical artery has the shear stress wall shear stress of 0.5 Pascal, and it can go up 10-fold and even 100-fold when you go into a stenosed artery. Now, we need a vessel that reacts to shear stress. And for the past 10 years, we have been developing these strange-looking liposomes. These are self-assembled liposomes, and the thing is they're made of non-natural phospholipids. Now, a natural phospholipid vesicle liposome is spherical. That's because you have 2 layers of lipids, and they float on top of each other. But with chemical tricks and physics tricks, we actually manipulate these membranes so that they interdigitate. And now we have 2 interconnected leaflets, and if you make this into a 3D-dimensional object, you really stress the physics, and you have to break the membrane. You introduce defects. Now, these liposomes are made of the artificial pad, a di-1,3-diamido phospholipid. Natural phospholipids, when you make them, when you make liposomes, they're tight or they're not tight. The pad is the very first vesicle that actually has some selectivity to shear stress. When you let the liposome stand on a bench, it is tight. When you start to shake it, it will release the cargo. And that's why we hope to use it for treatment of acute myocardial infarction. Now, question is obviously, how does that work? How does the release work? And here Marcia did her PhD to really elucidate the mode of action. She did some microfluidic devices and put them into small angle X-ray scattering at the Paul Scherrer Institute. And what we see is that when we stress these vesicles, they go from interdigitated to deinterdigitated. So they go from a gel phase into a liquid crystalline phase, and they become leaky. Natural phospholipids don't change their geometry. Now, question is always, we're using non-natural phospholipids, are they toxic? And together with Janos Sebeny, we did a lot of tests for immune system activation. Here is one where we take a pig and we inject naked liposomes of 0.5 milligrams per kilogram of pig. These are consecutive injections. The medical doctors doing this experiment decided to take the whole load of liposomes we gave them and inject it in one go. So we injected a very high dose of 5 milligrams per kilogram. And what you see here on the y-axis is the pulmonary arterial pressure. That's an indication of the activation of the immune system. And you see there's no activation of the innate immune system. When you add chymosan, a bacterial cell membrane, obviously this triggers the immune system. And you have to compare that to doxorubicin. FDA approved Doxil. We have 100 times less, and here we have a pulmonary arterial pressure going up to 40, where we have really an activation of the immune system immediately. Do we have tachyphylaxis? When we inject one dosage, is the next dosage actually having the same effect? When we start with a low dosage of a third of the human therapeutic dose, we don't have tachyphylaxis, so the next dosage will have the same effect. If we inject a high dosage, there we actually see a tachyphylaxis, but that's 3 times the therapeutic dosage. And I also want to come to a nice paper that was presented by Mogimi last year, where he actually said, you know, it's strange, but spherical particles are more toxic than non-spherical particles. And this is shown here. This is the pulmonary arterial pressure going up, and here we have injected round spherical particles, and they're much more toxic than the non-spherical particles that we now are developing. So this is a quick overview, and I also already want to end. I thank all the co-workers that helped on the project, and thank you for your attention. And this leaves some time for questions. And if there's no question,